So today topic is phospholipid and triglyceride metabolism, right? You find very difficult to learn the structure of phospholipids, or you have already learned, or we will try to make it easy or simplify it with diagrams. Okay, so what are triglycerides? So just see diagrams first. What is glycerol? What is glycerol? Glycerol. Anybody? Ol means alcohol, right? Then 3 carbons are there and alcoholic group is attached. CH2, CH, OH, CH2, OH. When 3 alcohols are there, it is known as glycerol. 3 ols means 3 alcohols are there, 2, 3 carbons, it is glycerol, right? And what are TGs? TGs or triglycerides. Any other name? Triacyl glycerol. What are they? Tri means 3 acyl groups with glycerol, right? Those 1, 2, 3. It is glycerol, right? And triglycerides are triglycerides or triacyl glycerol. Triacyl glycerol. Glycerol is 3 carbons with 3 ols. That is our glycerol, right? This one. And triacyl. Tri means 3 plus acyl. 3 acyl groups are attached to glycerol. It becomes our TG, right? Just see. Right? This becomes our carbons you add. This becomes our triglyceride, right? And what are phospholipids? Phospholipids. What are phospholipids? All the phospholipids are made from a parent phospholipid, that is phosphatidic acid, right? And triglycerides in this where occurs? It occurs in liver and adipose tissue, and fatty acid and glycerol must be activated for the synthesis of triglycerides, right? And this is the proper synthesis, and it is very difficult, right? But we will simplify it later on. Carbons are there, 3 alcohols are attached. What is formed? Glycerol, right? When you add one acyl group and another acyl group and another acyl group, it becomes acyl plus glycerol is equal to triacyl glycerol. And if one acyl group is attached, suppose it becomes monoacyl glycerol. 2 acyl groups are attached, it becomes diacyl glycerol. And another acyl group is attached, it becomes our triacyl glycerol, right? Now you draw the structure. What is this? This is phosphatidic acid, right? When 3 ols are there, it is glycerol. When one acyl group is attached to glycerol, it becomes monoacyl glycerol. Further, one more acyl group is attached, become diacyl glycerol. When 3 acyl groups are attached, becomes triacyl glycerol, right? Triacyl glycerol is clear. But on the third group, acyl group, when phosphate is attached, it becomes phosphatidic acid, right? It is a parent molecule for synthesis of all the phospholipids, right? And first of all, I will start with the metabolism or synthesis of triglycerides, right? From where the synthesis of triglycerides take place, it can be started from two products, right? One is our glycerol and second is our glucose, right? We'll start with glycolysis. Hope you remember the steps now. Glucose 2, glucose 6 phosphate, then fructose 6 phosphate, then fructose 1,6 bisphosphate, and then what will you get? We have glucose molecule. By glycolysis, it can lead to synthesis of dihydroxy acetone phosphate, right? Glucose, hexokinase reaction, glucose 6 phosphate, then again glucose 6 phosphate, fructose 6 phosphate, and 1,6 bisphosphate, and finally we'll get dihydroxy acetone phosphate, right? And glycerol via glycerol kinase reaction. Kinase means we are adding phosphate, so what we will get? Glycerol 3 phosphate. And from glucose, via glycolysis we will get dihydroxyacetone phosphate on the fourth step of glycolysis, right? From these two molecules, we can lead to synthesis of both triglycerides and phospholipids, right? So we have now glycerol 3 phosphate, right? Then we add acyl transferase. It will gets converted into lysophosphatidic acid, right? We have added one acyl group, right? And then another acyl group is added by acyl transferase. What will become? It will become our phosphatidic acid, right? And these two, glycerol 3 phosphate and dihydroxy phosphate, can be converted. Which reaction? Glycerol 3 phosphate dehydrogenase reaction, right? And NAD is used and it is converted to NADH plus H phosphate, right? Whether we start from glycerol or glucose, glycerol via glycerol kinase reaction, phosphate is attached, we get glycerol 3 phosphate, right? In the same manner, glucose via glucose 6 phosphate, fructose 6 phosphate, 1,6 bisphosphate, and we get dihydroxyacetone phosphate. These can be converted to again glycerol 3 phosphate via glycerol 3 phosphate dehydrogenase reaction via NAD reaction, and glycerol 3 phosphate, when we attach one acyl group via acyl transfer, will get lysophosphatidic acid, and again another acyl group is attached, we get phosphatidic acid, right? After that, what will you add? Can be converted into which form? Phosphatidic acid. I discussed the structure. Glycerol, one acyl group is attached, becomes monoacyl glycerol. 2 and it is converted to diacyl glycerol via phosphatase reaction, right? We got diacyl glycerol. Just see the structures first. I am not completing the valency, you will know, okay? So this is our glycerol. 3 O's are there, it is glycerol plus When we attach one acyl group, it becomes monoacyl glycerol. Again another acyl is attached, it becomes diacyl glycerol. Again another acyl is attached, it becomes triacyl glycerol. Clear? But now we have two acyl groups, that is diacyl glycerol, and here we don't have a acyl group and we attach phosphate group. What will it become? It becomes our phosphatidic acid. Clear? Right? So we have glycerol 3-phosphate. We attach one acyl group, it becomes lysophosphatidic acid. Then another acyl group, it becomes phosphatidic acid. Phosphatidic acid because it has phosphate at third position, right? But now we are doing phosphatase. That means we are hydrolyzing the phosphate. So what will get? Diacyl glycerol, because this phosphate is removed and by water. So we'll get 1, 2, 3, 1 acyl, 2 acyl, diacyl glycerol. Right? Clear? Now see. Now we have diacyl glycerol. What will you do now? In the diacyl glycerol, you add another acyl transferase. What will get? Triacyl. That is how triglyceride is synthesized. Right? Glycerol to glycerol 3-phosphate via glycerokinase and ADP is added. Right? Glycerol 3-phosphate to lysophosphatidic acid, one acyl group is added via acyl transferase. Again one acyl group is added, it becomes phosphatidic acid. Phosphatidic acid has phosphate at third carbon and two acyl groups. Right? After that we do phosphatase reaction and remove the phosphate from phosphatidic acid. So here we are moving this phosphate and it becomes diacyl glycerol. Right? From diacyl glycerol, we add another acyl group by acyl transferase and we get our triglyceride, that is triacyl glycerol. Clear? And other phospholipids, how you can synthesize. And this phosphatidic acid and this diacyl glycerol can lead to synthesis of all the other phospholipids. From this mechanism, we synthesize both triglycerides and phospholipids. Now we start with phospholipids in detail, right? So just see, any two can come on the stage. Are there? What is this? Glycerol, right? 3 O's are there. It is glycerol. With glycerol, I am attaching one acyl group. What will it become? Monoacyl glycerol. When another acyl group is attached, it becomes diacyl glycerol. Again, another acyl group is attached, it becomes triacyl glycerol. Right? Now, I am removing this acyl group and attaching this phosphate. What will it become? Phosphatidic acid. Right? 3 O's are there, it is glycerol. When one acyl is attached, it becomes MAG, monoacyl glycerol. Another acyl is attached, it becomes diacyl glycerol. If I attach another acyl, it becomes triacyl, that is our TG. Right? But at third position, we attach phosphate, it becomes our first phospholipid, that is phosphatidic acid. Right? From this molecule, we synthesize all other phospholipids. Right? Just see. So this is the complete structure of phosphatidic acid. Right? See, monoacyl, diacyl, triacyl. Right? At third position, we are attaching phosphate instead of acyl group, it becomes our phosphatidic acid. From this molecule, we can synthesize all the phospholipids. Okay? So this is parent molecule for phospholipid, that is phosphatidic acid, right? When we attached choline with it, choline, it becomes gas, phosphatidic acid plus choline, phosphatidyl choline, first phospholipid is formed, right? We have this phosphatidic acid. How is it formed? First we have 3 O's, that is glycerol. We attach one acyl group, becomes monocyl glycerol. 2 acyl group attached becomes diacyl glycerol. Another acyl group becomes triacyl glycerol. But at the third position now we have attached a phosphate, green one. It becomes phosphatidic acid, that is our first phospholipid, right? Now this phosphatidic acid is the parent molecule. We attach different groups and we form different phospholipids. We have phosphatidic acid and now we are attaching lecithin with it. Lecithin is found where? Lacithos means Greek word egg yolk, yellow part, right? It is present in egg yolk. When we attach phosphatidic acid and choline, it becomes lecithin, that is phosphatidyl choline. First phospholipid is okay, right? Now we have again phosphatidic acid. We attach ethanolamine. What is ethane? Methane, ethane, 2 carbons are there, and amine group is that becomes ethanolamine. Methane, ethane, amine. Ethanolamine is there, and now we are bonding phosphatidic acid and ethanolamine. What will get? Cephalin or phosphatidyl ethanolamine. Second phospholipid ban gaya. Clear? Okay. Now we have again phosphatidic acid. We are attaching serine. So what will become? Phosphatidyl serine. Remember one thing, we will start from glycerol. Glycerol. 3 carbons are there and 3 O's are there, that is our glycerol. One acyl group is attached to glycerol, it becomes monocyl glycerol. Again acyl group is it becomes diacyl glycerol. Third acyl group is it becomes triacyl glycerol. In the third position, when we remove acyl group and attach phosphate, it becomes phosphatidic acid. From this molecule we can form all the phospholipids, right? Now phosphatidic acid plus serine, it becomes phosphatidyl serine. We started with lecithin, phosphatidyl serine, and now serine, right? Now it's done. Now see, we have phosphatidic acid and this is phosphatidyl ethanolamine. What's the difference? This is phosphatidic acid. Structure is okay with you? Okay. Now normally we attached, hold you, amine, ethanolamine, it becomes phosphatidyl ethanolamine, and it is phosphatidyl ethanolamine. See the difference? Can you see the difference between those 2 phospholipids? Any guesses? We have ether linkage here in phosphatidyl, and this is phosphatidyl. It will come in multiple choice, pakka, 100%. Phosphatidyl and phosphatidyl. Then we have ether linkage at phosphatidic acid, carbon 1. It becomes phosphatidyl ethanolamine. Clear? And what is left? Phosphatidic acid is clear to all of you. 3 O's are there first of all. 1 acyl, diacyl, and finally phosphate becomes phosphatidic acid. Then we attach choline, it becomes lecithin, that is phosphatidyl choline. When we attach ethanolamine, becomes cephalin, that is phosphatidyl ethanolamine. Inositol is attached, becomes phosphatidyl inositol, and when serine is attached, becomes phosphatidyl serine. And in plasmalogen, this one we have ether linkage, it becomes plasmalogen or phosphatidyl ethanolamine group wala, right? Now the next is cardiolipin. Cardiolipin क्या है? You are familiar with structure of glycerol now? What is glycerol having? 3 OH group and 1 phosphatidic acid is attached, it becomes, right? But when we have to attach to glycerol, 1 phosphatidic acid at carbon 1 and 1 at carbon 3, it becomes our cardiolipid. See the name, diphosphatidyl glycerol, right? I am the glycerol molecule in the center. At carbon 1 I am holding hand with this 1 phosphatidic acid. At carbon 3 I am holding hand with this phosphatidic acid, right? But it becomes diphosphatidyl glycerol. Diphosphatidic acid with glycerol in center, it becomes our cardiolipid, right? So I repeat or not? Tell me. Just start from the beginning. So we started from glycerol. Glycerol means 3 O's, right? We attach 1 acyl group. It becomes monoacyl glycerol. We attach another acyl groups, it becomes diacyl glycerol. Then we attach another acyl group, it becomes triacyl glycerol, right? But instead of third acyl group I am attaching now phosphate. It becomes phosphatidic acid. Clear to all of you, right? Now we start from again. Phosphatidic acid. When we attach choline to phosphatidic acid, it becomes our lecithin phospholipid, right? And when we attach ethanolamine, it becomes phosphatidyl ethanolamine or saline, right? And when we attach serine with phosphatidic acid, it becomes phosphatidyl serine. And when we attach inositol, another group, it becomes phosphatidyl inositol. What is its use? It is involved in action of hormones like vasopressin, oxytocin, right? And plasmalogen. What is the difference between phosphatidyl ethanolamine and plasmalogen? We have ether linkage at carbon 1 with phosphatidic acid. Phosphatidic acid is as such there, ethanolamine is as such there. But we have ether linkage, then it will become phosphatidyl, not DYL, it is DIDAL, ethanolamine, right? Then last molecule was, we have glycerol in the center and 1 phosphatidic acid is attached, and we attach 2 phosphatidic acid, 1 at carbon 1 and 1 at carbon 3. It becomes our last one, cardiolipid, and it has antigenic properties, right? Sphingomyelins we will discuss in next class. Today we will finish only phospholipids, right? Synthesis of triglyceride is okay. Tell me, are you able to remember? We started from glucose and glycerol. Glycerol is converted to glycerol 3-phosphate via kinase reaction, and glycerol 3-phosphate we attach 1 acyl group via side transfer, it becomes lysophosphatidic acid. Again another acyl transfer, it becomes phosphatidic acid, right? Phosphatidic acid we use phosphatase reaction and water is removed, it becomes our diacyl glycerol. And diacyl glycerol has 2 acyl group. Di means 2 acyl group. When we attach another acyl group, becomes our triglyceride, that is triacyl glycerol, right? So for triglyceride synthesis is okay. Now we start with phospholipid synthesis. First we start with synthesis of lecithin and cephalin. So what is lecithin? I told you lecitos in Greek means egg yolk. The yellow part of the egg is richer source of this phospholipid, right? And it has a very good role in fetal lung maturity. आपको क्वेश्चन डाला गया था एफए में, इसमें से आया था, केमिस्ट्री में है, राइट? एंड डाइपाल्मिटोल lecithin is important phosphate that act as a surfactant in lungs, and lysolecithin is formed by removal of fatty acid either at C1 or C2, right? This is cephalin, phosphatidic acid plus ethanolamine. We will get our ethanol, nitrogen base with cephalin regard to base. Now we have phosphatidyl choline. This basic molecule is phosphatidic acid, and when choline is attached becomes phosphatidyl choline. And when ethanol minus attached becomes our phosphatidyl ethanol amine. Two structures are there. Then the third one is phosphatidyl inositol. We have inositol group attached. And what is its function? Important for signal transduction across membranes, right? And action of certain hormones like oxytocin and vasopressin mediated through phosphatidyl inositol. And this is inositol. This basic molecule is phosphatidic acid, and when inositol is attached, it becomes phosphatidyl inositol. Then serine, with phosphatidic acid plus serine will get our phosphatidyl serine. What is the function? It is involved in apoptosis or programmed cell death. This is basic phosphatidic acid, and we are attaching serine, it becomes our phosphatidyl serine. And last was plasmalogen. We have glycerin molecule. No. C1 we have ether linkage. Phosphatidic acid is as such is there and ethanol minus is there, but we have ether linkage at carbon 1, it becomes our plasmalogen or phosphatidyl. Previous one was PIDYL. This is PIDYL. Multiple choice आएगा PG में पक्का, ठीक है? Then cardiolipin, we have glycerol in the center, and on C1 we have one phosphatidic, and C3 we have another phosphatidic acid, becomes our cardiolipin. What's the function? It was first isolated from heart muscle, and two molecules of phosphatidic acid held with glycerol, and it is essential component of mitochondrial membrane, and its decreased level results in mitochondrial dysfunction, aging, hypothyroidism, cardioskeletal myopathy, also known as Bach syndrome, right? And it has good antigenic properties as well, right? So this is the synthesis of phospholipids. So we'll start from phosphatidic acid. Earlier we started from one was glucose, another was glycerol, right? Glycerol led to synthesis of glycerol 3-phosphate via glycerokinase reaction. Glucose via glucose 6-phosphate, fructose 6-phosphate, 1,6-bisphosphate will get dihydroxyacetone phosphate via steps of glycolysis, right? And glycerol 3-phosphate was converted to which product? Monocyl glycerol, that was phosphatidic acid, then phosphatidic proper, and then we converted into diacyl glycerol and triacyl glycerol, right? So we start with glycerol 3-phosphate, and it is converted to first phosphatidic acid. Hope you remember the structure of phosphatidic acid now. I will draw here. First we have 3 carbons, 3 O's are there. That is our, this molecule is glycerol. We attach one acyl group, we attach second acyl group, 1, 2. This is our diacyl glycerol. We attach here phosphate, and it becomes our phosphatidic acid, right? Now we are here at phosphatidic acid. We will synthesize all the phospholipids from this, right? How will you proceed? We have first 2 pathways, right? In one we add water and phosphate is removed, right? Phosphatidic acid is there. The structure is in front of you: diacyl glycerol and phosphatidic acid. Now we are adding water and phosphate is removed. Okay, so we will remove phosphate here. What will get? 1,2 diacyl glycerol. We'll get here 1,2 DAG. 1,2 diacyl glycerol. Are you able to carry it or not? So we have phosphatidic acid, monocyl, diacyl, triacyl, glycerol was there, diacylglycerol and phosphate is attached, becomes phosphatidic acid. Now we have removed phosphate via water and becomes 1,2-diacylglycerol. Then in another reaction we add CTP and phosphate is released, we'll get CDP diacylglycerol. Difference between two reactions, only phosphate is removed, we'll get the 1,2-diacylglycerol. And in this we have attached CDP, so we'll get CDP diacylglycerol, right? And further it has two pathways. We add inositol here. Hope you remember the structure of inositol. Let me show you. This was inositol, the 6-membered ring with phosphatidic acid, right? So we have CDP diacylglycerol and we are attaching inositol. What will become? Phosphatidal inositol. One phospholipid is formed. What are the functions of phosphatidal inositol? It is involved in the action of certain hormones, vasopressin and oxytocin, right? Another molecule, we add glycerol, 3-phosphate and CMP is released. What will get? Phosphatidal glycerol, 3-phosphate, from which we'll get finally phosphatidal glycerol, and one finally we'll get cardiolipin. What are the functions of cardiolipin? It was first isolated from heart muscle, so the name is cardiolipin, and it has antigenic properties, cardiolipin. Another function, it is involved in mitochondrial membrane permeability and role in mitochondria as well, right? So when we have phosphatidic acid, 2 phosphatidic acid attached, so we'll get our C, glycerol and cardiolipin diphosphatidal glycerol. How? See, what was this molecule? 3 O's are there, it is our glycerol. This is carbon 1, this is carbon 2, this is carbon 3. On carbon 1 we are attaching 1 phosphatidic acid, complete molecule. And on carbon 3 we are again attaching 1 phosphatidic acid. Diphosphatidic acid, glycerol. Diphosphatidic acid, glycerol in the center. Di means 2, phosphatidic acid, glycerol. It is our cardiolipin. Clear? Be alert, it is very important. You will get minimum 5 marks question out of this, right? So don't sleep in the class, be alert. Starting from glycerol 3-phosphate via glycerokinase reaction. From glycerol 3-phosphate we get our phosphatidic acid. Phosphatidic acid is a parent molecule from which we form all the phospholipids. Via first reaction we are adding water and moving phosphate. When phosphate is removed from phosphatidic acid we will get 1,2-diacylglycerol. And when CDP is attached with phosphate, we will get CDP diacylglycerol. In this molecule, when we attach inositol, we get phosphatidal inositol. This is another phospholipid is used in hormonal actions of vasopressin and oxytocin. And when we attach via glycerol 3-phosphate, another glycerol, and we'll get cardiolipin. In the center we have glycerol, 2 phosphatidic acids are attached, 1,2-diphosphatidic acid, glycerol. This is our cardiolipin. Another phospholipid is formed. It has antigenic properties, isolated from heart muscle, and plays a role in mitochondrial membrane. Right? Now another phospholipid. Acid, and it can also be converted into 1,2-diacylglycerol, but we attach here. I told you about choline. CHOLIN. What was choline? Again, it is present in yellow part of the egg. It is one of the very good phospholipid that is important for lung surfactant, right? So when choline is attached to 1,2-diacylglycerol, we get our phosphatidylcholine, right? How will you remember the structure? Okay. Again, 3 carbons are there, 3 O's are there. We have now which molecule? Glycerol. One acyl group. What is we having? Diacylglycerol. We attach phosphatidic acid. What will it become? Sorry, phosphate. I am attaching phosphate. Sorry, really sorry. What will get? Phosphatidic acid. And here we are attaching another molecule, that is choline. What becomes? Phosphatidylcholine. Another thing, lecithin. Mnemonic LPC. Lecithin is phosphatidylcholine. Choline is present in yellow part of the egg, that is egg yolk. Vision को भी नहीं आ रही है, very bad vision. Right? Now we have phosphatidylcholine. We have phosphatidylinositol. We have cardiolipin. Right? Anything left? 2 are left. Right? I will draw here. Phosphatidylethanol amine. Phosphatidylethanol. Phosphatidic acid plus ethanol amine. How? 1 carbon CH2, methane, ethane, amine. This is our ethanol amine. Ethane amine हो गई ना? तो when phosphatidic acid plus ethanol amine, what will get? Phosphatidylethanol amine, right? And here in phosphatidylethanol amine, we attach its reaction, we attach serine, and ethanol amine is replaced, we'll get phosphatidylserine, right? They are interconvertible, and when serine carbon dioxide is removed, it is again converted to phosphatidylethanol amine. So approximately we finished this phospholipids. Just one thing left. What is LCAT? Dinesh Puri sir was examiner of mine, and he asked me the second question. First I told you kinetic pathway, hope you remember, and this was the second question about LCAT. LCAT has very important role. Oh, one thing is also this left. We discussed the structure of lecithin. What was the mnemonic? Lecithin was LPC. Lecithin is phosphatidylcholine. L for lecithin, P for phosphatidyl, C for choline. LPC, right? So what is LCAT? It is from lecithin. It is lecithin cholesterol acyl transferase. Right? It has very important role in cholesterol metabolism, and it is synthesized in liver, and it is associated with ApoA1 of HDL. What is HDL? Good cholesterol, right? And LDL, bad cholesterol. So it is associated with HDL. So what will be the good function? It will help in reverse cholesterol transport and it will help in decreasing the cholesterol level of the body, right? The reaction is lecithin plus cholesterol. It will attach to cholesterol and form lysolecithin and cholesterol ester, right? The above reaction is responsible for reverse cholesterol transport mediated by HDL. HDL is good cholesterol, right? But why good? Because it has ApoA1 component of this lecithin. It will attach with cholesterol. Where it is present in the body, it will move and transfer via reverse cholesterol transport. So the good functions of HDL are mediated through this LCAT or lecithin cholesterol side transferase, right? Now one thing left is degradation of phospholipids. So degradation of phospholipids, phospholipase that break down phospholipids, lipase, phospholipid is a lipid, lip is lipid, AS means that break down the lipid is phospholipase. And we have different type of phospholipase, that is A1, A2, C and D. All has different roles and they target a different part of the phospholipid. The A1 will target here at acyl group 1. A2 will target at acyl group 2. And C will target at phosphate group and D will target at the base. So you find it difficult, but you will get short multiple choice from this. They will be giving 4 phospholipids and it will be phospholipases A, B, C and D, and multiple choice A1 will act at, you will be given choices base, phosphate or something else, but A1 will act at one acyl group, A2 will act at second acyl group, C will act at phosphate group and D will act at the base. What was the base? This one molecule is phosphatidic acid and the base is maybe it's ethanolamine, maybe it is lecithin, maybe it is inositol, right? So base is when target is activated by phospholipase D, right? And there are some separate roles as well. A1 specifically cleaves the fatty acid at C1 position of phospholipid, right? And later is acted upon by lysophospholipase B to remove second acyl group, right? And A2 where at C2 position of phospholipids, right? And A2 is present in which substance? It is present in snake venom and bee venom. They are highly rich sources of phospholipase A2, right? And C that cleaves the bond between phosphate and glycerol of phospholipids. And D removes hydrolyzes the nitrogenous base. Okay. Now see the diagram. A1 is acting at fatty acid 1, A2 at 2, C act as phosphate group, and D act at the base. So we discussed LCAT as well. So any doubts left? Class is over now.